Okay, photosynthesis, where plants make their energy, and basically where the energy for everybody else is made as well. It's done with this little formula here, and it pretty much represents what's going on in photosynthesis. But it's not quite as simple as it looks here. There's a complex series of reactions that happens to make this possible. They all happen inside of the chloroplast, which is inside of a plant cell, inside of the plant. And yeah, it's pretty small. Let's get a closer look at that. So what we have here is everything that makes up the chloroplast, and all of it is important. Let's start with the membranes. There are two of them. The inner membrane here, and also the outer membrane. They just kind of keep this whole thing together. Another important thing is the stroma. That is essentially the liquid that fills up the whole thing. One of the most important things in the chloroplast is the thylakoid. On its membrane is where most of the reactions that make photosynthesis possible happen. Also, the thylakoid space is super important to this. So we've got most of these parts here, but what do you call it when you have a stack of those thylakoid things? You call that a granum. And then you may have noticed these little pipes connecting them. Those are called the lamella, but that's not as important. On that thylakoid membrane, you have the light-dependent reactions of photosynthesis happening. The most important part of them happens in the photosystems, which look like this. And what this is a whole bunch of chlorophylls, these circles. The white ones are chlorophyll B, and the black one is chlorophyll A. Of course, they're actually green. They're not black and white. And what they do, so white ones, chlorophyll B, absorb sunlight and pass its energy on to chlorophyll A, where an electron is excited with the energy and passed on to an electron transport chain. This is basically why everything in this cycle is possible. So moving on, where do those electrons come from? Well, they come from the splitting of water on a protein attached to the bottom of photosystem 2, which, annoyingly enough, is the first photosystem in the whole chain of things. What happens here is the water is split into 2 hydrogen ions and oxygen. The electrons from the hydrogen are given to the photosystem, where with sunlight they can be excited and passed on down an electron transport chain. In this electron transport chain is a hydrogen pump, and what it does is it takes a little bit of energy from that electron and uses it to pump a hydrogen ion from the stroma into the thylakoid space. This is important because along with the splitting of water, it builds up a concentration gradient. We'll get to that in a second. So the electron continues down an electron transport chain to photosystem 1, the second one in the whole system. Anyway, at photosystem 1, more sunlight is used to excite the electron, which passes it down an electron transport chain. And this time it goes to a protein called NADP+ reductase. Here, NADP+ and 2 hydrogen ions, along with that electron, are used together to make NADPH, which is important because it is essentially an energy transfer molecule, and its energy can be passed on to the next part of this whole photosynthesis thing. Anyway, onto that hydrogen ion concentration gradient I was talking about earlier. Because there's more hydrogen ions inside the thylakoid space than outside of it inside the stroma, they naturally want to leave the thylakoid. And to do this, they have to pass through ATP synthase. And when they do this, a little bit of their energy can be used to combine ADP and a phosphate to make ATP, which is a form of readily usable energy for cells. It is also used in the next portion of photosynthesis. And now on to that. The light independent reactions, or the Calvin cycle. It happens inside the stroma instead of anything to do with the thylakoid. And what happens here is an enzyme called rubisco, there's 3 of them, are combined with 3 CO2s to make a unstable 6-carbon molecule. It makes 3 of them, of course. And because it's unstable, it then turns into 6 3-carbon molecules. They can't really be used for anything at this point, so energy from ATP, which turns into ADP, is used to rearrange it to make a slightly different 3-carbon molecule. Again, there's 6 of them. So that one still can't really be used to do anything in the cell, like make sugar or ATP or anything. So, some energy from NADPH, which turns into NADP plus and those hydrogen ions, is used to change its structure once again, to make 6 3-carbon molecules. These ones are called G3P, and 2 of them can be used for making glucose. So what happens next is 3 of them leave the Calvin cycle, and 5 of them continue on to rubisco regeneration. Once the cycle happens twice, you'll have 2 G3Ps here, which can combine to make 1 molecule of glucose. Now back to those 5 remaining G3Ps. They take some energy from ATP and turn it into ADP, and in doing this, they can be rearranged into rubisco again, which allows the whole cycle to happen again.